Okay, so this is our presentation about cyclin synthesis drives the early embryonic cell cycle, which is a paper by Murray and Kirshner in 1989. And we were assigned with analyzing figure 2C. So why is cell cycle research important? A little bit of background here. So cell cycle research is really important for cancer research and also embryonic development research. Cancer is caused by overactive cell proliferation, too many cells growing at once. So research into what compounds, what genes, what factors can control the phase transitions of cells can really shed insight into what exactly is causing the overactivity of the cancer cells. So to understand what drives cell cycles, it can be helpful to analyze what exactly is happening in each stage of mitosis. So we're going to start with interphase. So this section from G1 through S through G2 is known as interphase. And this is basically the period between cell divisions or cytokinesis. It's basically when the cells will copy their DNA into two sets of identical chromosomes. So as you can see here, this is just growth and preparation for DNA synthesis. The DNA replicates. It's basically just preparatory for the main stages of mitosis. So next, there's going to be prophase. And this is where the chromosomes are going to pair up and begin to extend to the opposite poles of the cell. And then next would be metaphase, where chromosomes line up at the sides of the cell and the mitotic spindles attach to the chromatids, basically getting ready to pull them apart. Next would be anaphase right here. And this is where the sister chromatids are pulled apart towards the separate poles of this newly forming cell or newly forming cells. And then finally would be telophase, which is where a new membrane forms around the chromosomes, and this is going to create the new nuclei. The cells are going to pinch down the middle, and two daughter cells are going to form. This is also known as cytokinesis. So in this paper, one of the main factors being analyzed was something called maturation promoting factor, or MPF. And it's basically what MPF basically is it's a complex formed by cyclin and CDK, which stands for cyclin dependent kinase. And H1 kinase is the kinase that's mainly related to MPF activity. So what H1 kinase does is it will phosphorylate a protein such as histones, which are needed for mitosis. And histone is a protein that aids in the folding of chromatin fibers during chromosome condensation. So MPF is going to appear at multiple stages of the cell cycle, but mainly during the transition of interphase to mitosis. So the main question would be, is cyclin sufficient to drive cells through the phases of mitosis? Like what factors, what compounds are required to onset mitosis or to drive cells from interphase into cell division? Chris, go ahead.
So based on the idea we learned from this paper, when trying to demonstrate the biochemical event occurring in this extract, we look into the change of some special protein. Mitosis promoting factor is a complex formed by cyclin B binding to this CDK1 protein, which is it has this three place to do the phosphate. And when the MPF complex began to dephosphorylate. The this complex acts as a regulator that enable the cell to enter the mitosis stage. When cell division process to the earlier telophase, cyclin B is degraded and MPF drops to the undetectable level. Since the function of histone is to compose the structure of chromosome, the function of histone kinase is to phosphorylate the corresponding histone to participate in the relevant activity of the extract or either cell before prophase. Therefore, in the extract, by assaying its concentration level to explore the cell cycle under the activity of H1 kinase, the relationship between this and the MPF. If the activity of H1 kinase increase before the breakdown of the nuclear membrane, at the end of the interface, it can reflect its correlation with the entry of cell into mitosis. And at the end of the time, DNA replication was monitored using DNA binding fluorescence staining. According to the completion of this running on the scale test result, 20 minutes and 60 minutes was the time of 2-cell cycle and the nuclear membrane breakdown. At this point, the test result of the histone kinase and MPF were in the highest level. And therefore, the MPF and histone kinase assays in figure 2C can reflect this periodicity of the two mitosis related to complex during the incubation process of the extract. So for the experimental option, in order to get to know the correlation between those complex, we need to detect the activity of them by assigning their activity at different times to compare the either morphology under the microscope and the Gill analyzing this cycling protein after the fluorescence staining. When we take the sample, we dilute this into half in the EB solution. To make the EB solution, preparing the concentration at 80 millimole per liter potassium and 15 millimole magnesium chloride and 20 millimole potassium EGTA solution. And with the final fixed pH at 7.3. And when we have this mixed solution, they analyzed this in the frog egg cell. When adding in the amount of MPF, that can make a half of germinal vesicle breakdown in the egg cell. That would be recorded as a unit of MPF. And when taking the sample, diluting this into 1/50 and put it into the EB solution. After they added about 10 microliter of 1 milligram per milliliter histone, 1 millimole per liter ATP, and a trace of the phosphate label ATP. Separate on 5 to 15% GAL gradient. And the activity of H1 kinase was expressed by calculating the improperate phosphate. And for the result, MPF activity and the H1 kinase activity spike at point of nuclear breakdown, undetectable by earlier telophase. Cycling appeared to activate kinase activity that induce appearance of MPF. MPF appear to be regulated by the cycling as a compound. Yeah.
So there are a few criticisms that I have for this experiment. So first of all, you know, this obviously, like a lot of science experiments, this wasn't perfect. So first of all, this experimental setup of extracting cytoplasm from electrically activated eggs is a relatively new development. The researchers actually designed this approach like just for this experiment, so it isn't exactly the most well-studied technique. And then secondly, as with like all science experiments, reproducibility is really important. If other scientists can't or won't reproduce your experimental setup, then the experiment can't really be validated very well. Since you can't really have 100% accurate data with running only like one experiment, you're going to need a few other goes to see if there was some bias going on in the first one. There just needs to be reproducibility. Secondly, there are a few discrepancies in the graph, as you can see here with the yellow arrows, between MPF activity and the H1 kinase activity. The researchers note that this may be due to the high threshold of the MPF assay, but I think the experiment could still be reproduced to maybe validate the accuracy of this graph. Finally, this isn't, this obviously isn't their fault, but it's a relatively old experiment coming from our point of view now. This doesn't necessarily invalidate the results at all, but I think new technology has been developed, and that could potentially affect the results of this experiment if it was being conducted with new methods and today's knowledge as well. And finally, connection doesn't necessarily equal, or correlation doesn't always equal causation. Just because cyclin and MPF may appear at similar times, they could be correlated together, but it's hard to really say if one actually causes the other, or what the relationship is there. So again, reproducibility is really important there. There's the references.